In last lecture, we learned that the virus, their morphology, their characteristics, and their properties, and their symmetries also, right, Shweta? We learned symmetries also.
Yes.
Then after that, we have started the viral, so certain virus have the capsid is covered with viral envelope. This viral envelope is also covered. Might be I will repeat once again. This envelope is covered around the capsid, which will give the more extra protection to the virus, and this is the envelope. This envelope is derived from or formed from lipids, and this is essential to release the host cell during the budding of the cell surface. This envelope is made up of or formed with a glycoprotein in nature, or this envelope is formed with glycoprotein in nature, or it is made up of by naturally by glycoproteins. So this envelope is susceptible means it will get harms from ether and chloroform and any other detergent. The examples of this are envelope viruses are herpes virus, hepatitis B virus, and HIV virus. These are the examples of envelope virus. Okay. So see now non-envelope viruses means non-envelope, it means the viruses which doesn't possesses the any covering around the capsid. Those are categorized in the form of non-envelope virus. These are also called as a naked virus. These are naked virus. It means that capsid is the resistant to. It means the lipids, chloroform, ether, or detergent doesn't brings the harmfulness to non-envelope virus. Means there is no action which seen with these agents towards the viruses. Next parameter is that is a papillomas. What is meant by papillomas? It is a matured virus particle, or a virus which is attained a particular age, that is the papillomer. That is a papillomer. It is a mature virus particle which consists of a glycoproteins, which consists of a spikes. What it consists? They consist of spikes. Spikes means a rod-shaped antennas around their capsid. Those are spikes of their envelope. Those spikes are called as papillomas. Understood? A virus may have more than one type of papillomas. Means papillomas are there. More than one different types papillomas are there. For example, we'll take an example of virus containing a papilloma, that is the influenza virus. It carries only 2 papillomas. It means there are papillomas are there over the envelope, but the number of papillomas varies from virus to viruses. These papillomas brings the hemagglutinin and neuraminidase. heme agglutination reactions and neuraminidase enzymes may be present in the papillomas. Okay. See the functions of papillomas. What are the functions of papillomas? Obviously, papillomas are antenna rod shape found over the envelope of virus. They helps to attach the host. Means the papillomas is helping to the virus to attach the a host cell, because papillomas have the particular sites which are matching with the receptors of the host cell. Then if the virus matches with the receptor's host cell, then papillomas are initiates the entrance of viral nucleic materials towards the host cell. Means attaching towards the host cell can be initiated by a papilloma by matching with the receptors with respect to of host cell to the virus. When the receptors are matching, and then papillomaries start the initiation of attachment, and after initiation of attachment, after the attachment, the release of genetic matter towards the host cell is possible. This can be achieved with the help of papillomas itself. The receptors, suppose these papillomas, when these are binds to the red blood cells of the human, there is an heme agglutination, and they will bring the enzymatic activity with the enzyme of neuraminidase, which gives the neuraminic acid to the host cell, glycoproteins. These papillomas have antigenic properties. Means these are possess the antigenic properties. See, this is genetic matter we will see in the a virus, which is covered with a capsid. So these are the only viruses called as naked viruses. Same virus is covered with the envelope, then we will call it as a envelope virus. You are watching in the screen. Hello.
Yes.
Apart from that, we are watching the green type spikes around the envelope. These are called as papilloma. What these are? These are all papillomas. This is non-envelope virus. This is envelope virus which carries the papillomas in their envelope. See, this is an example of a this is a virus. It is influenza virus. This is an envelope virus which carry a capsid with a genetic matter. Around that there is an envelope. Envelope carries the spikes on their envelope. It may be a spikes are two types. These are H spike and this one is N spike. So this is what about the spikes enveloped virus. These spikes are helpful for attaching towards the host cell and penetration of these nucleic materials or genetic materials towards the towards the towards the host cell, then the spikes may bring the heme agglutination reaction, as well as these spikes brings the enzymatic activity, neuramidase enzyme, which will brings the uramic acid within the cell of host cell, and uramic acid which will bring the reactions inside the human RBC cell. So this is what about the capsid envelope, why capsid containing genetic matter of the virus. This is naked virus. This is covered with the envelope with the spike. This is the envelope virus. Then spikes, human influenza virus containing H and N spikes over the envelope. So this is what all about the information of envelope and non-envelope viruses. We are moving to next slide. Viral nucleic acids. See, already we know viruses contain either DNA or RNA, but not both. These DNA or RNAs brings the replication of viruses, means the new proteins or formation is possible and new viruses are formed. In a host cell, this is the genetic matter is transferred from through the spikes and formation of a new proteins in the host cell. This DNA or RNA, they may be a single-stranded or double-stranded. They might be circular, see, like this. They may be a single-stranded or they may be a double-stranded. They might be a circular or they may be a linear. They may be a segmented or non-segmented also. According to nucleic acid present in the virus, DNA, according to the presence of genetic matter in the viruses, they are classified on the basis of DNA viruses and RNA viruses. Okay. We will move to next slide. See, this picture representing the presence of a envelope, capsid, circular network-like genetic matter. This is what about envelope virus. This is the influenza virus. This is HIV virus with spikes we are watching on the envelope of virus. See, these are the examples of envelope viruses. See, this one is also a genetic matter with a capsid and spikes we are watching in this slide. So now we will able to tell you it is spikes containing, envelope containing viruses are human virus, HIV virus are the example of envelope viruses. See now how the viruses are multiplied. How the viruses are get multiplied. See, the virus multiplication is possible by a lytic and lysogeny of lytic and lysogeny methods are there for the multiplication of viruses. It means that there is no doubt viruses are multiplied in the host itself, where they are multiplies and they will get forms in the host. See, the virus, it doesn't have its own machinery to reproduce. Their progenies in their host, that's what they are dependent to other host. Then what are the steps are involved in viral multiplication? Keep in your mind always, in both lytic and lysogeny, these certain steps are common. That is, adsorption is also called as attachment. It means that in adsorption and attachment, the spikes will come nearer to the host cell. Spikes will come to host cell, and if the receptors are matching with respect to the host cell, then spikes will adsorb or attaches over the host cell. Then second step, it is a penetration. Penetration means the ejecting out of the content of genetic matter of the virus towards the host cell. It is a penetration. Then uncoating. Uncoating, it means the genetic matter which is covered with a layer, it will get uncoats into the cytoplasm of the host cell. Then biosynthesis of a viral genetic matter in the host cell with the help of false proteins. With the help of false proteins, biosynthesis will start. The viral genomes are get biosynthesized. See, this adsorption, penetration, uncoating, biosynthesis are common for both lytic and lysogeny. Then maturation in lytic, and then there is a bursting of host cell and release of progeny in the environment. This is about the release of progeny. So these are the common steps. There is adsorption, penetration, uncoating, biosynthesis, maturation, and release of the virus into the host. These are the steps we will see inside the viral multiplication. Here, lytic and lysogeny. These are these two steps involved in viral multiplication. These are the common steps in both lytic and lysogeny. Maturation and releases we will see with respect to lytic. So one can one can reverse the process. That is called as induction. What it is? Induction or induce. We will transfer the lysogenic state to lytic or lytic to lysogenic. This can be possible. We can be achieved with the help of ultraviolet radiation. Certain chemical like H2O2 are used to transfer the state of lytic to lysogenic. This is called as induction or inducing the change in the state. This can be possible. So we will learn these topics, these matters in further slides also. Okay. Slide. See, adsorption or attachment. Already we know this virus, which is comes nearest to the host cell for binding towards the host cell. Before binding towards the host cell is called as, that virus is called as virion. And this virion comes closer towards the host cell and attaches to the receptors of the cell surface. For example, an influenza virus which is having a spikes which comes towards the RBCs, it will bind the receptors like glycoprotein, hemagglutinin binds, specifically celiac acid residue. This is a glycoprotein receptor, and influenza virus binds with the host cell. In case of HIV virus, that is also consists of a spikes, binds to glycoproteins of GP120. It's an action ligand and binds to CD4 cells of T lymphocyte. Means this is the binding site of virus, it is HIV virus of T lymphocytes. CD4 T lymphocytes are target cells on which the HIV virus will get binds on the surface with the help of glycoprotein GP120 acts as a ligand or receptors for the binding site. So means the respective spike will bind at the respective receptors of a particular category cells itself. If the virion falsely binds to the host, if the receptors are not matching, then it will get aborts automatically. There is no attachment proper and there is no release of viral DNA or RNA into the matrix of host cell. Okay. Now, penetration. After binding the spikes over the host cell, there is a next step is starts or initiates. The receptor mediated endocytosis. Receptor mediated endocytosis. Mostly non-enveloped viruses, receptor mediated endocytosis will say. In receptor mediated endocytosis, we will see in non-enveloped viruses. And in enveloped viruses, the envelope will fuses with the cellular membrane and delivers the nucleocapsid and genome directly into the cytoplasm. This is what all about the viral multiplication, that is by adsorption and penetration. So we will see the next topics, next points in further class. Okay.